Current technologies allow us to extract bacteria from the skin, modify them in the laboratory, and engineer them to do what we want. We like to call this Skindev, as in skin devices, because they will be like microscopic living machines working on our skin. Skin diseases are a big health problem worldwide. There are more than 3,000 skin conditions, and 1 out of 3 people are affected by them. Some of them are lethal, like melanoma, while others are socially impairing, like eczema. Traditional treatments for skin diseases are limited. For example, for atopic dermatitis, doctors generally recommend the frequent application of creams and topical corticoids for acute inflammation. But those treatments have been the go-to for 60 years. Recently, new treatments with monoclonal antibodies have been approved, but they are still not a complete solution. They are expensive and they are reserved only for those with severe symptoms. Nowadays, many laboratories around the world are able to take bacteria from the skin, grow it in flasks, and apply it to people as probiotics. But we are the first to go beyond that. We want to genetically modify these bacteria to generate what we like to call skin devices.
Skindev is a project that aims to create new paradigms for diagnostics and therapy of atopic dermatitis. We use some bacterial devices that are able to sense the beginning of the disease and will be able to produce therapeutic substances against the disease. And this project is formed by teams of 4 different countries, Germany, Netherlands, UK, and Spain. And we are very thankful to the European Innovation Council to support this project.
So here at Imperial College London, we focus on developing computation models to better understand the dynamic interplay between the skin, its immune system, and microbiome. It helps us identify the targets and markers for skin disease, and also to better understand how to modify C. acne to design skin devices, Skindev, as new diagnostic and therapeutic tools.
Here at the University Hospital Düsseldorf, we are a team of active dermatologists continuously facing the current problems of skin disease. With our clinical view, we will help to develop and fine-tune the skin devices in a way that is best for all patients.
Our skin is the boundary between the external world and our body. We will produce a therapeutic platform that is capable to sense the first signs of the disease and react with producing therapeutic molecules that should help in bringing down the inflammation associated with the disease.
C. acnes is a very particular bacterium. It is the most abundant bacterium in our skin and it lives deep in the hair follicles. This is why we believe C. acnes is the perfect chassis to develop our skin devices. Our long-term vision is to take natural C. acnes from the skin, to modify it in the lab by introducing DNA that will provide it with new capabilities, and then to put it back to the skin of patients so that it can detect and treat disease. The advantages of using C. acnes are many. C. acnes populates the skin, which is the interface between the external world and us, so that it can be used to detect signals that are either external from the environment or internal from our body. Also, it is important to note that C. acnes are alive in the skin, which means that they can monitor disease continuously and also produce beneficial molecules over long periods of time. For these reasons, we believe that C. acnes could be used to provide health monitoring and treatment 24/7. Since these bacteria live in the follicles and not at the very surface of the skin, they can deliver molecules to the deepest part of our body, and also they are more protected from chemical or physical insults that may affect the skin. Finally, once we develop a functional skin device, it can be very easily produced because bacteria can be grown in bioreactors in large amounts, resulting in low production costs.
Skindev is a highly multidisciplinary effort covering different objectives in computational biology in the United Kingdom, synthetic biology in the United Kingdom and Spain, and dermatology in the Netherlands and Germany.
Here, also at Imperial College London, we work together with a team at University Pompeu Fabra in Barcelona on the actual engineering of Cutibacterium acnes. We take the wild bacteria and we develop tools and methodology to be able to domesticate them.
Here at the Radboud UMC in Nijmegen, the Netherlands, we create skin in the lab. And we try to recreate the patient's skin to the best of our ability. And we do this by integrating the computational analysis together with experimental data and with the patient data itself. Thereby we create a high-tech platform in which we can test the brand new skin devices for their efficacy.
With the collaboration of all the partners, we hope to develop a new generation of skin therapies and diagnostics. In the end, it all comes down to benefiting from the natural adaptability of C. acnes to the skin, and also to providing these bacteria with new capabilities to help them help us.
We believe that the microbial devices that we are creating, they can generate a new paradigm for skin care and dermatology in general. During this project, we will transform this idea into a reality.